What is immunohistochemistry in cancer diagnosis? Imagine trying to find a tiny needle in a haystack. That's what doctors do when they look at tissue samples from a biopsy to figure out if cancer is present. Sometimes, just looking under a microscope isn't enough to tell the difference between normal and cancerous cells. That's where immunohistochemistry, or IHC, comes into play. It's a special lab test that helps identify specific proteins on or inside cells to give a clearer picture of what kind of cancer they are dealing with. The process starts with a tissue sample taken during a biopsy. The sample is prepared on a slide and stained with tiny antibodies that are designed to stick to certain proteins called antigens. These antibodies are like tiny detectives, searching for specific markers that are unique to different types of cancer cells. When they find their match, they produce a color change, usually a brown or red stain, that can be seen under a microscope. This makes it easier for pathologists to identify the cancer cells and understand their characteristics. This technique is especially helpful when the initial biopsy results are unclear, or when the doctor needs more detailed information to decide on the best treatment. For example, in breast cancer, IHC tests can detect hormone receptors like estrogen and progesterone, as well as the HER2 protein. Knowing whether these markers are present helps doctors determine if hormone therapy or targeted drugs will be effective. It's also used in other cancers to find specific proteins that can guide treatment options. IHC is usually done after the first biopsy if the results are not definitive. It's a reliable method that adds important details beyond what standard tissue examination can show. This extra information helps the medical team create a personalized treatment plan tailored to the specific features of the cancer. For families and caregivers, understanding this process means knowing that the diagnosis is thorough and that the treatment options are based on precise information. It's a key step in understanding the nature of the cancer and how best to fight it.